Today we're going to talk about karyotyping of induced pluripotent stem cells, what it is and why you should do it. So an outline of today's talk. First of all, we're going to talk through the karyotyping process. I'm going to cover an introduction to karyotyping, discuss examples of abnormalities, discuss how we report abnormalities, mention changes of abnormality frequency, and discuss some other detection methods. So first of all, the karyotyping process is a bit more involved in than some other tests you may have come across. As first of all, we need to perform cell culture. Once the cells have been cultured, we can harvest them using standard cytogenetic techniques. Then we can produce our slides. We can then G-band our slides using TRIPSA and GIMSA, and this produces a characteristic banding pattern across our chromosomes. And then this enables us to analyze our chromosomes. So on the first image, this is representative of what you might see on low power down the microscope. You can see we've got round interphase nuclei, and we've also got some short metaphase cells. The second image is a close-up of one of these metaphase cells. And the third image is a screenshot of an imaging software called Cytovision, which is one of the ones available, and we can use this to produce a karyogram. So at CellGS, we do some of our analysis down the mic, but the imaging software is really useful when we want to produce a karyogram, or when we're karyotyping particularly complex cells, or when we're karyotyping non-human species. So this is an example of a normal male karyotype. This is a karyogram that we might send out to customers. This is normal because it has 46 chromosomes, which is the standard amount. And also because we can see from the banding pattern that there are no structural abnormalities present. So this is a male cell. This is 46XY. When we're karyotyping, we're checking for numerical changes, either gain or loss of chromosomes. We're also looking for structural changes, rearrangements, and it's the banding pattern that allows us to do this. If you look on the right-hand side, you'll see a close-up of two chromosome 1s. And these have been banded using G-banding. The trypsin is a protease enzyme, and then the GIMSA stains. So what we have is we have these dark bands, which are relatively gene poor, which have not been very digested by the trypsin. The light bands have been more digested by the trypsin, and they're gene rich. So this is quite a nice cell. We would normally look at at least 20 cells for an analysis. And this is probably, and each cell would have all the chromosomes checked, and there would be an assessment of the quality. So this cell is probably a quality 4 to 5. This is because the number of bands, or the resolution present, is quite high. We can see on 18q that there are 4 distinct bands, and that makes this at least a quality 4 cell. Typically for embryonic stem cells or induced pluripotent stem cells, quality 3 might be more typical, but it does vary a lot. It can vary either within the sample, dependent on the sample type, and it can also vary according to how well the sample has been processed. So starting with numerical abnormalities, it's quite obvious that there are too many chromosomes here. This is an example of polyploidy, which is 92XXYY. So this is basically a doubling up of the normal chromosome complement of a male cell. And actually all the chromosomes here, although there are too many of them, are structurally normal. There are no structural rearrangements. Polyploidy actually represents an inherent genetic instability within cells. So it's actually quite common for polyploid cells to have a very complex karyotype, which means that there can be 5 or more rearrangements or gains or losses within a single cell. So when we're karyotyping, each cell can just have a single abnormality, or it can have multiple abnormalities present. Within cell culture, up to about 10% of cells being polyploid is considered normal. We report them when we see at least 15 to 20%, and that's especially if they are tetraploid, which is 4N or higher. This is another example of a numerical abnormality. This is trisomy 12, so one of the more common ones seen in ESCs and iPSCs. You can see here that there are 47 chromosomes rather than the usual 46, and that we've gained an entire copy of chromosome 12. So it's important to remember with these gains and losses of chromosomes or these structural rearrangements that actually they can result in gain or loss of thousands of extra gene copies within a genome. But here we've identified that the gene that's important is the nanog gene, and that maps to the short arm of chromosome 12. It maps to 12p13.31. And this gene encodes for a homeoprotein, which is required for pluripotency sustainment. Gain of 12p13.1 appears to reduce the ability of cells to complete hepatic progenitor differentiation. So overexpression of nanog and also the gene GDF3 seems to result in a feedback loop that keeps cells in a pluripotent state. So cells remaining in a pluripotent state and not fully differentiating, the reason why that's of interest to researchers is because in clinical use, this actually leads to risk of teratomas. So I've discussed some types of numerical change, and this diagram shows some of the common types of structural rearrangement that we see within chromosomes. It doesn't demonstrate all of them. There are also isochromosomes and ring chromosomes, amongst others. But to start with, we've got duplication. That's straightforward. That's just duplication of material within a chromosome. Those can either be just a straightforward tandem duplication, or you can also get inversion of the material as it duplicates. The second abnormality is inversion. That's just where the order of the chromosome banding, as we would see it, reverses. Deletion of material from chromosomes is often seen. We can have insertion, where we get material from one chromosome inserting itself into the banding pattern of another chromosome. And we can also get reciprocal translocations. This is where material from one chromosome exchanges with material from another chromosome. And these can either result in a balanced form of translocation or an unbalanced form. So, an unbalanced translocation, we have an example here. This is also demonstrating overall gain of 1q material, which we frequently see in iPSCs and ESCs. The ISCN at the top is telling us that this cell has 46 chromosomes. It's a male cell. It has a derived chromosome 1 from a translocation between two different chromosome 1s, where material from the short arm of the long arm of one chromosome 1 has translocated onto the short arm of the other. What this results in overall is trisomy, or 3 copies for that chromosome 1 material, that chromosome 1q material, and you can probably see that from the banding pattern. So the gene implicated here is MDM4 on 1q32.1, and this is known to regulate and interact with p53. The pathway is affected. It raises the threshold for cell apoptosis. There are also other candidate genes that have been put forward for this region, but MDM4 is the main one. It's quite recently been published that cells that have gain or duplication of 1q material show increased proliferation both in vitro and in vivo, with decreased cell differentiation again. This is another example of an unbalanced translocation, another structural abnormality. This time we have a derived chromosome 15 from a translocation between one copy of 15 and one copy of 17. Material from the long arm of chromosome 17 has translocated onto the bottom of chromosome 15. So overall, this results in trisomy for 17q material, and you could have this through any number of different translocations. It's just manifested in this way here. The gene involved here is BRCA5 or survivin, which is located at 17q25.3, and there's also STAT3 located at 17q21. These both regulate apoptosis, and when they're overexpressed, apoptosis is inhibited. Another type of structural abnormality that we commonly see is isochromosome 20q. So 20q gain overall is common in iPSCs and ESCs, but this form, this isochromosome, is particularly common. What you have here is loss of the 20p material and gain of 20q. You've basically got two copies of 20q forming a mirror image around the centromere. This results in overexpression of a gene called BCL2L1, which is located at 20q11.21. An overexpression of this allows cells to escape from apoptosis. Papers have been published that show that gain of this region appears to prevent neuroectoderm differentiation, and that's due to altered TGFB and SMAD signaling. So we have another structural abnormality here. It's duplication. It results in the same 20q gain as I discussed on the previous slide. So the reason to show you this slide is because we have duplication of the 20q11.2 band, and it's quite subtle. This is about at the limits of resolution that we can expect to see on G banding. The limit of resolution for karyotyping is probably about 10 megabases overall for the type of cell that we're looking at. What you can see here, this stretched out white band, that's actually amplification of the general region. And to see whether you had duplication of that particular candidate gene, that BCL2L2, you would need to use a confirmatory method, perhaps such as SMP arrays, which we offer, and we would mention this in our report. So, as I mentioned, we also do mouse chromosomes. The normal number of chromosomes for mice is 40, and in this cell we have 42 because we have trisomy for mouse chromosome 1, and we have trisomy for mouse chromosome 11. The trisomy for chromosome 11 is particularly interesting because it leads to a third copy or gain of 11q-E2, and that's actually synthenic to 17q25 in humans. And as we've discussed, that results in gain of BRCA5 gene. When we're karyotyping, we can see either all normal cells, we can see all abnormal cells, or we can see a mixed cell population. And that's what we refer to as mosaicism, when we have two different cell populations within a single sample. So to start with, we can have a mix of normal and abnormal cells. So on the left, we have a normal male cell, and on the right, we have a cell that has trisomy 12. We can also have two or more abnormal clones with unrelated abnormalities. So on the left, again, we have our trisomy 12 cell, but on the right, we have a cell that has a deletion of the long arm of chromosome 18. We can also see evidence in the form of mosaicism that an abnormal cell is acquiring more cytogenetic abnormalities. So again, on the left, we have our trisomy 12 cell. On the right, this cell also has trisomy 12, but in addition, it's also gained material onto the long arm of one copy of chromosome X. So this is a sign that the karyotype is evolving in complexity. So we don't get a vast range of iPSC and ESC cells at CellGS because often we're repeatedly analysing the same cells after increased passage number for our customers. However, as I've shown, we do see most of the common ones. And here we have just an overview of the most common main recurrent cytogenetic abnormalities. So for ESCs, we have gain or trisomy of chromosomes 1, 12, 17, 20, or X. We have duplication of the long arm of chromosome 1. We have partial gain of chromosome 12, partial gain of 17, and duplication of 20q. For iPSCs, we tend to see fewer trisomies overall, perhaps with the exception of trisomy 8 and trisomy 12. We see more amplification of the region 1q31.1, amplification or deletion of 17q to 1.1, and amplification of 20q11.21. For mice, we see trisomy of 8 and 11, and also deletion of the regions 10qb and 14qc to 14qe. So overall, this year, for the 300 or so samples, both iPSC and ESC that we've karyotyped so far at CellGS, we find about 70% of them to have a normal karyotype and about 30% to have an abnormal karyotype. And of the abnormal ones, the vast majority are accounted for by abnormalities involving the same 6 chromosomes, chromosome 1, 8, 12, 17, X, and 20. But we always have to be on the lookout for other changes. So, for instance, there are deletions of 18q, there's polyploidy, and so forth. So when it comes to reporting the abnormalities that we find, we follow a few general rules. Now, standard clinical practice for reporting cytogenetic abnormalities, you would normally expect to see structural abnormalities at least twice, and you would hope to see numerical abnormalities seen 3 times to confirm that they are a genuine finding. However, at CellGS, we do things slightly differently because we report some recurrent abnormalities even if we see them only in a single cell. And that's because we know that cells containing these abnormalities always outcompete the normal cells and take over the culture. We record all abnormalities, but we do not always offer interpretation. And that's for cases such as there may be novel abnormalities seen where there is no published data to allow interpretation of what this would actually mean for the cells. And also, we recognize that some chromosome changes are potentially transient, and we would advise perhaps a follow-up karyotype of subsequent passages. It's really, really important to realize not all cytogenetically abnormal cells will have the potential to change the properties of a culture or induce an increase in the proliferation rates of cells or of cell survival. What is interesting is that over time, anecdotally, we've find this in-house, but there do appear to be changes in frequencies of cytogenetic abnormalities. And the best published data I found on this has come from the University of Wisconsin in collaboration with their Stem Cell and Regenerative Medicine Center. So they looked at 7,300 samples between 2009 and 2016. And in these samples, they came across about 1,300 chromosome abnormalities in about 1,200 cultures. And that will be because some of these cultures will have had more than one abnormality. So between the, within the time period, they noticed that there was an increase in 1q gains from 12% to 27%. They noticed an increased frequency of 20q gains from 6% to a maximum of 43% in 2014. And they noticed a decreased frequency of 12p gains to 21% from 60% over the studied period. And this was consistent across both iPSCs and ESCs. This has implications for culture methods and also detection methods. So there are factors that affect genetic stability of cultures. And within the time period studied by the poster authors, methods for derivation and culture of pluripotent stem cells have changed. What we do know about genetic stability of cultures is that there is a positive but not complete relationship between increased passage number and the increased number of genetic aberrations. We know that the low physiological O2 concentrations and addition of small molecules such as nucleosomes can reduce DNA damage and increase stability. We also know that gentle cell handling techniques increase or decrease DNA damage. For instance, for disaggregation or passaging methods, mechanical methods are generally considered gentler than enzyme methods. So increased increased cell culture density, suboptimal culture conditions, increased selective pressure. And what we know is that chromosome changes are basically considered a response to increased selective pressure. With regards to detection methods, if these types of abnormality change over time, you would need to consider if you are using a targeted detection method such as PCR or FISH, that your assay was designed appropriately. When we talked about this in-house, we thought it was quite interesting that the number of 20q abnormalities, the detection of them had increased, because it's actually one of the more difficult cytogenetic abnormalities to detect. When we were talking about how the frequency of 12p has decreased, we wondered whether this is because 12p is often, gain of 12p is often manifested in the form of an isochromosome 12p, and that tends to be something that's present at quite low mosaic levels. We have noticed that with increased time, fewer 60-cell analysis have been performed and more 20-cell analysis. As a rule of thumb, we know that if you analyze 20 cells, you would expect to detect a mosaic clone of just 14% with 95% confidence. Whereas if you increased your analysis up to 50 cells, you would expect to see a clone size of just 6% with 95% confidence. So we do have customers that do choose to karyotype 20 cells, but we also have many who choose to karyotype 40 or even 60, just to increase the confidence with which they can exclude mosaicism. So it's important to mention that there are different methods for detection and for checking the genetic integrity of your cells. So there's quantitative PCR and FISH. So that's polymerase chain reaction and fluorescent in situ hybridization. These are both regarded as targeted testing methods. The pros of them are that they are cheap and that they're easy to interpret. The cons are you really need to consider which primers you're using, whether they reflect the abnormalities that you hope to detect. They also rely upon selection of stable reference genes. Now, historically, these have been housekeeping genes in cells, but a lot of these were characterized decades ago. And there's some evidence that within a very dynamic cell culture with high cell proliferation, such as you might find in iPSCs, that they may not be as representative as we'd hope. Because you're using targeted testing of a few key genome areas, it does mean that normal results can be potentially misleading because it doesn't exclude abnormalities elsewhere in the genome. Both of these methods have quite limited ability to detect mosaicism of just 20%. So there's SMP arrays, which are single nucleotide polymorphism arrays. We offer these at CellGS. They have a higher resolution of just 200 kilobases compared to perhaps 10 megabases with G banding. And they're good for detecting unbalanced rearrangements. The cons of this method is they cannot detect mosaicism, so they cannot detect balanced rearrangements or polyploidy. Where they do detect an unbalanced rearrangement, they can advise as to the overall imbalance of genetic material, but they can't give you the mechanism or the chromosome rearrangement of how that has occurred. With SMP arrays, data analysis and understanding of CNVs, copy number variants, is required. So basically, there are CNVs throughout the genome, and they have varying significance. Some are known to be benign, some are known to be pathogenic, and then there are others which are considered to be variants of unknown significance. So when we're interpreting those results at CellGS, we make sure that we're following the latest US and European guidelines, especially with regard to handling of any copy number variants. Next generation sequencing or whole genome sequencing have the advantage that they can detect changes down to the single nucleotide level. They can detect copy number change and structural variants. However, although the price of them has come down, they are still considered expensive. The significance of single nucleotide variants is often unknown. There's limited published data. And really, if you're going to look at the change, when you interpret your data, you really do need access to the parental cell line DNA to establish what changes are acquired throughout your cell culturing process and what might have already been there within the parental DNA. Optical genome mapping is perhaps the newest technology. It has a resolution of just 15 to 30 kilobase pairs and can detect balanced and unbalanced structural rearrangements across the whole genome. The cons are that it cannot detect polyploidy, and it requires very complex data analysis. So finally, we come to karyotyping, which is a long-established method. And because it's a long-established method, that means that not only are the results readily interpretable, it often means there's a lot of published material about how to interpret any findings that you come across. The main advantage of karyotyping is that it can detect very low level mosaicism down to 1 in 20 cells. And detection of mosaicism is actually really important because it's been published in the literature that normal cells can be induced to behave more neoplastically through cell-to-cell contact with cells that have gain of either 12p or 20q. Also, apoptosis of normal cells can be induced through cell-to-cell contact with abnormal cells. So it's important to get a true reflection of what cell types you have and whether you have a mixed cell population within your culture. So because karyotyping looks at all of the chromosomes and the entire genome, it can detect unbalanced and balanced rearrangements. It can detect polymorphic variants that occur throughout the human population, and it can also detect polyploidy. Also, karyotyping does allow us to see random breakage that can occur in chromosomes due to clastogenic agents within the cell cultures. Of course, the main cons of karyotyping is that it is relatively time consuming because it requires cell culture. So it has quite a long turnaround time, and it also requires specialized analysis. And the resolution is 10 megabases. Despite these cons, karyotyping does remain the gold standard for confirming genetic stability within PSCs. So why do we analyze stem cells? So the white paper that came out in 2018 listed a number of mandatory QC attributes for iPSCs. And at the bottom, you'll see that under genetic fidelity and stability, we have residual vector testing and karyotyping. So residual vector testing just is to make sure that your vector has not integrated into your host cell genome. And of course, karyotyping we have discussed. So as of papers published this month, karyotyping is still listed as an exemplar method. And the white paper and this paper from September 2022 specify that at least 20 cells should be analysed by a qualified cytogeneticist. Single cell abnormalities may require further investigation when they're seen, i.e. you may need to increase analysis to 50 cells, or you may wish to consider further analysis of later passages. It's also important to consider how often you should be karyotyping your cells, as we know that some of the cytogenetic abnormalities such as 20q gain can confer proliferation advantage within as few as 5 passages. So the guidance for this probably comes from the European Centre for Validation of Alternative Methods. They had a series of workshops in 2007 and published a paper in 2012, and they recommend karyotyping every 10 passages. So recommendations for analysis of iPSCs. For clinical use, it's recommended to karyotype a minimum of 20 cells every 10 passages to check for chromosome abnormalities that have significant impact on cell growth. For research use, the advice is less clear. However, Isu et al. Proposed that iPSC genome quality should be monitored at derivation and then at least every 12 weeks, i.e. approx every 36 cell divisions. This is seen as a sort of midway between doing nothing and the amount of analysis required for clinical use, and is just an acknowledgement really of the cost implications. And finally, SNP analysis or array genome hybridization is a useful tool to identify changes at a higher resolution than may be afforded by G-banding. So finally, the cytogenetic services offered at CellGS. Well, we've been offering a karyotyping and SNP array service since 2018. We process approximately 700 samples per year. Our staff are fully qualified. We're experienced both within NHS laboratories and also other clinical laboratories overseas. And part of that experience means that we understand the limitations of the various genomic tests that are offered and can offer experts advice in their interpretation. Our staff follow good laboratory practices. For instance, cell transfer steps are all witnessed and countersigned, and also we have a good paper trail for documenting how we process our samples. We can offer a fixed cell service or a live cell service, so you can either send your cells to us and ready for slide making, or you can send us live cells and we'll harvest them for you and follow through for you. The analysis of cell numbers can be adapted to suit the clients. As I mentioned, we usually do a minimum of 20 cells, but that can be to 40 or 60 as required. We offer human and mouse karyotyping routinely, and we can offer other species available on request. And we're always really happy to hear from customers and to answer any queries. So we can be reached on the email address below. And I think that's all. So thank you very much for your time.
That's great. Thanks, Lotta. So we have a few questions that have come through. So if anybody else has any questions, feel free to put them in the Q&A box. The first one that came in, unfortunately, there's no name, but the question is, why are there differences in the recurrent chromosomal abnormalities seen between ESCs and iPSCs? Why do ESCs have a different set of chromosomal abnormalities compared to iPSCs?
Well, there's actually quite a lot of overlap in that both iPSCs and ESCs, we tend to see the same chromosomes involved. We send so 1, 17, X, 20, 12, etc. The differences are probably due to the way the cells were derived. So obviously the ESCs have been taken from an embryonic blastocyst, sorry, whereas the iPSCs have come from cells, that somatic cells that have been fully differentiated, and then they've been reprogrammed back to a pluripotent state. So there is some research that suggests that even if you use transcription factors to bring cells back to a pluripotent state, they may actually have some memory of sort of from imprinting of the state they were from before. So that might explain the difference.
Okay, thank you. So the next question from Joydeep Dutta is, when you found mosaicism of numerical aberrations, what phenotypic characteristics were observed?
So we expect trisomy for chromosome 1 and trisomy for chromosome 12 to result in increased cell proliferation. So when we harvest our cells for culture, because we use trypsin to lift the cells from the flask, and we produce a sort of cell suspension to make the slides, we can't actually link back cells to individual, say, cell colonies that might have been behaving in a certain way, that might have shown that they were in particularly proliferative, I guess.
Okay.
So that might be one where we would actually need, when we received fixed cells rather than the live cells, it would be that the customers might actually be able to tell us how they noticed their cells were behaving, or the phenotype that they had that was unusual.
All right, thank you. So the next question is from Joshua Clayton, and Joshua is asking, do you see any trends in the types of abnormalities based on reprogramming method used, e.g. episomal versus Sendai virus for iPS cells?
We don't, but that's probably because we don't actually get that information from our customers. So if we did, we might be able to sort of draw some conclusions from what we tend to see with various customers, depending on the reprogramming method they use. And it might be interesting to look into.
Yeah. Okay, and another one from Joydeep. What percentage of cells show chromosomal abnormalities after 10 passages, for example?
It varies. Well, it would vary a lot because it's very dependent on how the on the cell culture conditions. There is a paper, and I cannot think of the name off the top of my head, but if we took their email address, we could forward it. But it does show you, it does give percentages for the percentage of different samples you might expect to be to have gained chromosome abnormalities after each sort of set point of passages.
Okay, that's great. So maybe if we look that up, we can share if that person, Joydeep, if you'd like to drop us a line, I'm sure we can get that information to you. So the last question I have, unless there's any more, but again from Anonymous, my team is working with macaque cells. Do you process these cells, and what difficulty do you face while processing non-human cell lines?
We have processed various primate samples, and we can karyotype them. The only issue we have with karyotyping is it tends to be a little bit more time consuming, just because we tend to image every cell and karyotype each cell individually, whereas for human cells, we're so familiar with seeing human chromosomes that we tend to just do that down the mic. So really, it's just the slightly more time consuming.
Okay, that's great. Well, thank you, Lotta. If there are no more questions, then we'll close the webinar here. So one late question just come in. Is there much from Alistair Fraser? is there much need for karyotyping in differentiated cells, brackets immune cells, for example? Regulators are looking for this.
Sorry, I didn't quite catch all the question. What type of cells was it?
So is there much requirement for karyotyping of differentiated cells such as immune cells? And Alistair says the regulators are looking for this. I guess it's things like CAR T cells for immunotherapy.
I guess what the regulators would, for the immune cells, they would be interested to see whether perhaps we were picking up some of the standard cytogenetic changes that are associated with some of the leukemia type analysis, because there are sort of particular characteristic changes of the various immune cells within leukemias. So it might be that they're looking to exclude, but I don't know of any particular recommendations for differentiated cells.
Okay, that's great. Well, thank you very much, Lotta. Great presentation, and thank you for everybody for joining us today. I hope you have a great rest of the day wherever you are, and if we can support you with your karyotype analysis in the future, we'll be certainly very glad to do that. So thank you once again. Bye-bye.